So I'm pretty confident if we went out to Hampstead High Street and we asked people if they'd ever heard of hepatitis, that the vast majority would say yes. So I think probably most people would have known something about what Mala was talking about. I think if we went out and asked people had they ever heard of primary immune deficiency syndromes, I'm fairly confident I wouldn't get too many takers. So primary immunodeficiency syndromes are inherited immunodeficiencies. So unlike HIV, which is an acquired immune deficiency syndrome, people are born with these conditions. And the work that I'm going to tell you about is a big focus for our immunology department here, and is not performed simply by my group, but is formed in collaboration. So the people who are involved in this, some of whom are here, are the clinical immunology team, and also Professor Dave Sampson's group, who is very much involved with helping us to understand the function of immune cells in these conditions. So I thought I'd start somewhat at the beginning. So this is a picture of your bone marrow, and if I was going to ask you where you thought your immune system was, probably if I asked you where your liver was, most people could point to it. If I asked you where your immune system was, that might be a bit trickier. So essentially our immune system is composed of our white blood cells, and these start, most white blood cells start life in the bone marrow, which is shown here. So this is a biopsy of a bone marrow. This is bone here. And if you have a roast chicken on a Sunday and you take the leg and you snap it, the dark bit in the middle is the bone marrow, and this is this bit here. And the bone marrow is dark because it's packed full of blood cells, and there are lots and lots of red cells in there which make it dark, but there are also very, very many white cells. And in fact, this is the area where most of your immune system or most of your white cells are made. And if we were to look in a bit more detail at what's in the bone marrow, the bone marrow has stem cells, and stem cells, of course, have become quite famous in recent years. They're in the media quite a bit, what stem cells might be able to do. But these are essentially a bit like parent cells that live in the bone marrow, or they can live in other tissues. But for the immune system, stem cells for the immune system are in the bone marrow. And these are a bit like parents that can give rise to different sons and daughters. And these sons and daughters give rise to grandchildren, and all of these cells are white cells of the immune system that are all slightly different from each other. Amala has spoken about these cells, NK cells, and T cells, but broadly our immune system can be divided into the cells on this side of the slide, the left side here, which are called myeloid cells, and these are the cells that are your first line of defense. So if you get an infection in your skin, these are going to be the cells that rush there to start to deal with the bugs that you've got. These cells on this side are actually much better at providing a memory, and that's why as we get older, children have lots and lots of infections, but as we get older our immune system learns, and these cells, which provide the memory part of our immune system, help to protect us from infections as we get older. So let's just have a think for a minute about what happens in an immune response. So this is a tissue, so let's say this is your skin, and you get an infection here in the skin. There's some inflammation caused by the infection, there might be some bugs here. And then into that area of inflammation come immune cells, and these are your rapid first line defense immune cells, and there are a number of different types here. This is called a neutrophil, we might have a macrophage, this is a dendritic cell, but essentially these are rapid responders. Once these cells start to respond, some of the specialized cells, like this dendritic cell here, will move into the lymph node. So when you get swollen glands, if you have tonsillitis, that's exactly what's happening. The rapid response cells are moving into the lymph node, and then they snuggle up with the other cells that are in the node, which are your T cells and B cells. So Mala was talking about T cells. And these are the ones that are going to learn how to deal with the bug and provide long-term memory for your immune system. And the cells must get very close to each other here, and I'll explain why in a moment. But the overall purpose of this is for all the cells to learn about the bug that you've been infected with and to produce things like antibodies, which are proteins. Yep, here we go, which are produced by B cells. And these are proteins which are released into the bloodstream that can help to protect you from infection next time. So for the next slide, let's just remember that when you get an infection, you have these three cell types mainly. You have this green cell, which is called an antigen-presenting cell, so that's picked up the bug. You've got T cells and you've got B cells, and the overall immune response, one of the major things is going to be to produce these antibody proteins. Okay, so this looks a bit complicated, but let's just break it down for a second. So this is the same kind of idea. So pretend we're looking in the lymph node here. This is the B cell that's going to produce the antibodies here at the end. And this B cell can only do this if it talks to a T cell, which talks to this dendritic cell or antigen presenting cell. And the way these cells talk to each other is through sensors on their surface. So the sensors on the surface of the T cell, we call receptors, talk to the sensors on the surface of the B cell, which are other receptors. And these are very important interactions for this cell to tell this cell to tell this cell what to do. And ultimately, this cell is telling this cell to tell this cell to make antibodies. So what happens when bits of the system are missing? This is when you have a primary immune deficiency disorder. And this looks a bit horrific, but some of you may recognize this. So this is a boy called David who has made primary immune deficiency a little less obscure than it would otherwise be, because there was a film made about this boy in the 1970s with John Travolta. And this boy here had a very severe form of immune deficiency where he was missing T cells and B cells. And if you lack parts of the immune system, whether it's T cells or B cells, you end up with infections. And you can get really abnormal infections. All of us get infections. Most of us don't have a serious problem with our immune system. But some people do, and they might get terrible warts like this, recurrent abscesses, this is an abscess on a neck, or terrible problems with viruses like varicella virus, this is chickenpox, that normally the rest of us would get over fairly quickly. So these primary immune deficiency syndromes are what we are interested in here in this department. And the most common immune deficiency that we look after is something called common variable immune deficiency. And mostly that's something that comes on either, can be in childhood, but often in adulthood. And it's called common. In actual fact, it's not very common. It's probably only got, probably in the UK, there'll be a few thousand people affected with this condition. It's called variable because although we call everybody with this condition common variable immune deficiency, every patient with this deficiency can be a little bit different. The one thing that's common to everybody is in all of these cases, these antibodies we were talking about, these proteins that protect you from infection, these are missing. Now, from the previous slides I showed you, there might be a number of reasons for that. In any case, if you have antibodies missing, the problem that you get is frequent infections. Some patients only get infections and some patients get other complications. And so even though we call everybody common variable immune deficiency, so the label on the bottle in some ways is the same for all the patients we see in our clinic, we know that actually that what we're looking after is a group of different disorders. And one of our big aims is to try and sort this out. Instead of calling everybody the same condition, try and work out why some people only have infections, why some people have lots of other complications. So in actual fact, try to define out what are the subgroups within this condition to try and make treatment better. So treatment really, if we're going to move forward for this condition, which can be serious in adults, needs to be a bit more personalised. So we're not giving the same treatment to everybody when actually the people with the same label have got probably slightly different diseases. So how are we going to address that? So I showed you this slide before, and this is just saying that at the end here, if you focus on these blue things, all the patients that we see with common variable immune deficiency fail to make antibodies. Now you can see from this slide, which is why we were going through it earlier on, that actually this could be because you have a problem in your B cell, it could be because you've got a problem in the T cell that has to talk to the B cell, or it could be because you've got a problem in this antigen presenting dendritic cell, which has to talk to the T cell to talk to the B cell. And we know already that some of the sensors which are on the surface of the cells here, if they're missing, we know they cause immune deficiency. Now the ones that I've circled in red are ones that we already know cause problems in humans. So ICOS deficiency, for example, we don't need to go into them. It's just to point out that a single molecule or a single sensor missing on the surface of the cell can give rise to a problem with antibodies. Now, we know a few of these conditions, but for most people who come to our clinic with common variable immune deficiency, probably less than 10% we actually know the genetic diagnosis for, actually probably less than 5%. And I think if we're going to move forward, we need to actually understand more about which bits of this pathway are missing so that we can understand for an individual patient, okay, you've got a problem with this or you've got a problem with that, that might help us to treat you better. Okay, next slide. Thanks, Lucy. So how are we going to do that? So this is something that we're doing with Professor Sampson's group. And essentially one thing that we want to do is test immune cell functions. If you remember back to that slide where I have the dendritic cell that talks to the T cell that talks to the B cell, we can measure, for example, the number of B cells. We can measure whether those B cells make antibody. We can look at things like whether or not the T cell and the dendritic cell actually stick together and talk to each other and whether those sensors interact. And we can look at whether all the cells that are supposed to be there actually are there. So by designing tests in the lab to try and understand, are all the immune cells there? Do they talk to each other? Are all those sensors on the surface working properly? We can try to divide out all the different types of common variable immune deficiency according to immune cell function. But we know that immune cells are controlled by the genes that are in the nucleus of the cells. This is a picture of a cell. This is the nucleus in here in green. And in here is the DNA, which is the control center for the cell. So we know that DNA is made up of lots of building blocks, and that DNA codes for most things about us. So what color our eyes are defined by what our genetic makeup is. How tall we are is defined by what our genetic makeup is. And also, actually, how our immune cells work is defined by what our genetic makeup is. And so if we were to take my genes or your genes and decode them, what we would be looking at is a very, very, very long string of, which is a bit like a computer code or a bit like a telephone code. So each single building block of the DNA is represented here by a different letter, and we have millions and millions and millions of these. Now we know from things like the Whole Genome Project that actually the whole human genome has been sequenced, so we know what these letters should look like in a healthy individual. So one of the approaches that we're taking with our immune deficiency patients is to actually sequence their entire genome and ask, do they have a normal genome, or are there some changes here in the genes? Now, if you took me and you took you, we're all going to have some changes in our genes, and we're not all going to be exactly the same. And one of the challenges for us as researchers is to try and work out whether this change up here or this change up here actually causes disease or not. But this is one of the approaches we're taking. So we're looking at the DNA, we're looking for all the changes, and we're trying to understand which changes here might be important for common variable immune deficiency, and which changes here might be important for the immune cell functions that we're measuring in the lab. So this is our kind of overall approach for our department for how we're looking at these immune deficiency disorders. We want to look at genetic information to understand what gene problems there might be. We want to look at immune cell function to understand whether we can divide out people based on which immune cells are working and which ones are not. And then obviously we look at how patients present. So do they present only with infections? Do they have infections with lots of other problems? And this all takes a lot of analysis that, as I said, is involving a number of groups. But our overall aim is to try and turn what is at the moment quite a complex condition, common variable immune deficiency, which is a whole ragbag of probably different disorders, and try and work out, is this actually a number of individual conditions, and can we find these individual conditions and devise treatments that are better for them? And we know that this approach it does work to some degree, because already there are new genes being described for immune deficiencies very regularly, but recently there was one described for CVID, and that particular gene defect results in a problem for which there is already a medication. And that medication has been trialled in a different condition, but that medication can probably be moved into clinical trials for those patients with that particular type of CVID quite quickly. So I think we do have proof of principle that this type of approach will work. If we can divide out what the different components of CVID are, we can probably generate better treatment for patients. Thank you.